In this short tutorial, we'll look at the ways that water-soluble and lipid-soluble hormones interact with cells and bring about a cellular response. Water-soluble hormones would generally include peptide hormones, protein hormones, or amino acid hormones, whereas lipid-soluble hormones are more likely to be hormones such as steroid hormones. Let's have a quick look at the key differences between these two kinds of hormones. With a water-soluble hormone, essentially the hormone, which we'll call H, or the ligand, will bind to a receptor embedded in the cell membrane. Since a water-soluble hormone will not be able to pass through the lipid membrane easily, so the hormone will have to bind to a receptor, probably a glycoprotein of some kind, embedded in the membrane, and in turn this will set off a reaction with a secondary messenger inside the cell, and we'll look at this in a little more detail in just a moment. And this secondary messenger will bring about a signal cascade, or a cascade of reactions that will ultimately influence the DNA, or the genetic material of the cell, which will bring about some kind of response. The key bit to realize here is that a water-soluble hormone cannot pass through the membrane, and so it must interact with a receptor that's embedded in the membrane itself. Alternatively, a lipid-soluble hormone, such as a steroid hormone, is capable of dissolving in the phospholipid membrane, and should be able to pass through. So in this case, the hormone will be able to pass through the membrane and will then interact with a receptor on the other side of the membrane, and this in turn will set off a series of reactions, a signal cascade, that likewise will interact with DNA to bring about a response. The key bit here is to realize that a lipid-soluble hormone, such as a steroid, can pass through the membrane and will interact with a receptor in the cytosol. Now let's have a look at each of these in a little more detail. Alright, let's have a look at how water-soluble hormones act in a little more detail. This diagram is a variation on one that you'd find in your Biozone manual, and so you should be able to find an equivalent there to follow. So essentially it's the same as we've talked about previously. So here we're dealing with a water-soluble hormone, let's say a peptide hormone, that will be travelling in the bloodstream, so we call this the first messenger. This hormone, or ligand, will bind to a transmembrane receptor. Now a transmembrane receptor just means a receptor that is situated across the phospholipid bilayer, across the membrane. So we can see that here. In this particular example, this transmembrane receptor is adenylate cyclase. And you can see that the hormone will bind to the receptor site here, which will activate the receptor. The activation of this receptor will in turn bring about the activation of a secondary messenger, in this case cyclic AMP, CAMP. So cyclic AMP is made from ATP, and this requires the action of the enzyme adenylate cyclase. So the hormone binds to the receptor. This initiates ATP being converted into cyclic AMP, which then in turn activates another enzyme, protein kinase, which then sets off a signal cascade. That is a series of biochemical reactions which ultimately causes the activation or deactivation of a gene on DNA in the nucleus. This in turn will bring about some kind of response, either the production of a particular enzyme or protein or the stopping of such a reaction. Again, the key thing to realise here with a water-soluble hormone is that the hormone is not capable of passing across the membrane, so it must interact with a receptor that's already in the membrane. Now let's have a look at how another variation in this will work using a G protein. This is another example of how a water-soluble hormone, say a peptide hormone, could interact with a cell. Instead of, in this case, cyclic AMP being involved, this involves a secondary messenger called a G protein. But essentially, the process is the same. So we have a receptor, a protein receptor, embedded in the phospholipid bilayer. We have what's called a G protein that is linked to the membrane. We can see that here, which has a molecule of GDP, guanine diphosphate, attached. Just a small correction there, GDP is guanosine diphosphate, not guanine diphosphate. So a molecule of guanosine diphosphate, GDP, is attached to a G protein, and in this form the G protein is inactive. So in this process, again, we have the peptide hormone, the signaling molecule, or the ligand, binds to the protein receptor in the membrane. This stimulates the G protein to lose GDP and replace it with GTP, guanosine triphosphate. This activates the G protein, which then moves along the cell membrane and then binds to another enzyme in the membrane here and activates it. This in turn then brings about a signal cascade, which ultimately leads to interaction with the nucleus and some kind of response through protein synthesis. After this process, the GTP is lost or used up, and GDP again binds to the G protein and inactivates it. And then the cell is ready for another reaction. So in this particular case, the primary messenger, the first messenger, is the signaling molecule, and the secondary messenger involves the G protein in here, which carries the message to this enzyme, which is then activated to carry out a signal cascade. Now let's have a quick look at how steroid hormones work. Okay, let's just expand a little on how a lipid-soluble hormone such as a steroid like cortisol might work. So again, we begin with the steroid hormone travelling in the bloodstream, but because it's lipid-soluble, it means that it can easily pass through the phospholipid bilayer, and it will then interact with an intracellular receptor. That is a receptor that's inside the cell, in the cytosol. So we will have a receptor here that's capable of binding to that steroid hormone. This will ultimately cause the activation of certain genes on the DNA, which then in turn will lead to an alteration of cell function through the production of a particular enzyme or protein in the cell. So in this case, we don't have a secondary messenger. The action is more direct.